When I started my research at UCLA, I thought every major component of the human cell had been identified. Wow, was I wrong. Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist. My passion is a common particle of the human cell called a vault. My goal is to explain what vaults are to anyone interested in science, nanotechnology, and health. You know, vaults live in a nanoscale world, so the first video I made was an explanation of nano and nanotechnology. If you haven't seen it, I've added a link below. Today I have three simple goals. First, I'm going to explain to you a bit about the history of cell biology and explain the difference between eukaryotic and prokaryotic cells. Second, I'm going to give you a brief tour of the components of a cell. It's mostly stuff you learned in high school. Some of this stuff is taught in the 7th grade. And finally, I'm going to tell you about my research, which led up to the discovery of a new cell component, the vault. Hopefully, I'll convince you that vaults are interesting and worth some of your attention. Cell biology really had its birth during the Great Plague of London, which ravaged from 1665 to 1666. It killed over 100,000 people. That was about 20% of the population. Far worse than COVID. You know, it would take over 200 years before it was determined that plague was caused by a bacterium. Hell, in the 1600s, we had no idea what bacterium were or that they even existed. It was during the plague that a talented English botanist by the name of Robert Hooke used a home-built microscope that magnified about 30 times. Not very impressive. Why, the camera in your iPhone can magnify more than that. Hooke focused his microscope on a tiny thin slice of cork from the bark of an oak tree. He saw a pattern of tiny rectangular holes in the cork that he said looked like walled compartments of a honeycomb. The little compartments reminded him of the cells that monks lived in. That's why Hooke called them cells. Around this same time, a Dutch scientist named Anton van Leeuwenhoek designed an improvement of the microscope by using double convex lenses mounted between brass plates. His microscope magnified objects an unheard of 300 times. Leeuwenhoek, who is now considered the first microbiologist in history, observed an abundant number of single-celled and small multi-celled organisms, which he called animalcules. These were swimming in a drop of rainwater. They were the very first scientific observations of amoeba and bacterium. From these profound observations made over 350 years ago, other important scientists added to the development of cell theory, an elegant hypothesis that cells are the basic unit of life. We now know that all living organisms are made from single cellular units, and that a cell is the smallest unit of life. There are only two types of living cells, prokaryotic and eukaryotic. Prokaryotic cells include all bacteria, like the common gut bacteria, E. coli. These cells consist of a single closed compartment surrounded by a membrane. They do not have a nucleus. And although prokaryotes are very important in biology, I'm not going to talk about them today. Rather, I want to focus on eukaryotes, which include all plants and animals. Eukaryotic cells contain a defined membrane-enclosed compartment for their DNA called the nucleus, and extensive internal structures. Remember in my previous video, I prepared a drop of my own blood and we viewed it under the electron microscope? I showed you this white blood cell, and I pointed out the two major cell compartments, the nucleus, this large area in the center of the cell, and the cytoplasm, the area around the nucleus. I'll use the analogy that the cell is like an independently functioning walled city. The main building materials for this city, the bricks and mortar, come in four major types: amino acids, sugars, nucleic acids, and lipids. Amino acids are the building blocks for proteins. Sugars are the building blocks for complex carbohydrates. And nucleic acids assemble into DNA and RNA. The lipids are oil-like molecules that self-assemble into membranes. These membranes surround and protect the cell, and they surround the individual compartments of a cell, which are called organelles. You know, when I was 18, I developed an interest in science. My early PhD training was as a biochemistry graduate student at the University of Michigan, and I worked in a lab that studied lipids. I want to spend a few minutes discussing the basic structure of a eukaryotic cell. I'll draw a human cell called a fibroblast. Fibroblasts are cells derived from the connective tissue of skin, and they can be grown in the laboratory. These cells are very useful to cell biologists. Here's their basic shape, and here's a scale bar. A fibroblast is about 15 micrometers across. Eukaryotic cells are surrounded by a membrane called the plasma membrane, which acts as a permeability barrier, keeping unwanted materials out of the cell. Usually near the center of the cell is the nucleus. Using the city analogy, I'd describe the nucleus as the central library of the cell, which houses all the instructions for making a cell. The instructions are written in DNA. The nucleus is filled with chromatin, which is a combination of DNA and DNA binding proteins, and a subcompartment of the nucleus called the nucleolus, where some RNA is made. The membrane that surrounds the nucleus is a double membrane that contains many specialized passageways. These are called nuclear pores. These pores regulate the movement of molecules between the nucleus and the cytoplasm. The outer nuclear membrane is contiguous with another compartment of the cell called the rough endoplasmic reticulum, or RER for short. RER is identified by these granules which decorate the membranes. Many of the cell's proteins are made in the RER. Near the RER is another compartment called the smooth ER. This is where many lipids are made. Usually near the ER is a stack of membranes called the Golgi complex. This is where newly made proteins are processed and sorted, kind of like the post office, except everything gets efficiently sent to the correct location. Another double membrane compartment of the cell are the mitochondria. These are the cell's power plants, where energy to power chemical reactions is produced. This is an electron micrograph that I borrowed from a paper published in 1955 by a cell biologist by the name of George Palade. Palade used the same stain I used to stain my blood, a metal that binds to membranes. And here you can see mitochondrion, these long oval structures labeled M. Here's one highlighted. The stain also binds to DNA and RNA, and that's why Palade was able to notice these very small granules labeled G here in the picture. Palade called them granules, but at the time they were referred to by others as Palade bodies or Palade granules. You know, later they were renamed the ribosome. And in 1974, Palade shared the Nobel Prize for the discovery of the ribosome. Ribosomes are those small granules that decorate the ER membranes, and they're also found free throughout the cytoplasm. Ribosomes are one of the most abundant particles of the cell. They're about 25 to 30 nanometers in diameter. Because they're not surrounded by a membrane, they're not classified as organelles. Ribosomes are actually machines that assemble amino acids into proteins. This is such a fascinating process that I'll go into it much more in a later video. One of my favorite organelles is the lysosome. This is the cell's recycling center. I studied this organelle for 5 years while I was a postdoctoral fellow at the NIH, also known as the National Institutes of Health. NIH is the largest federal research institution in the United States. The lab I worked in was right here. The next organelle I want to mention is the peroxisome. Peroxisomes are similar in some ways to lysosomes, as they detoxify some materials and they break down some lipids so they can be recycled. Rounding out the major components of this fibroblast are various small vesicles involved in transporting stuff throughout the cell, and a bunch of different cytoskeletal fibers that form networks and bundles and help organize the cell, determine cell shape, and participate in cell movement. These are the cell's highways and train tracks and bridges and more. So, with that crash course in the structure of the eukaryotic cell, I want to tell you about the early work in my lab that led to the discovery of what turned out to be one of the most abundant particles of the human cell. After my postdoctoral studies at NIH, I accepted an assistant professor position at UCLA in the Department of Biological Chemistry. Here's a photo of me around that time. Scary, huh? I started my own research program, continuing my studies on lysosomes. Specifically, I was interested in the enzymes inside lysosomes. 1 minute on enzymes, so we're all on the same page. Enzymes are usually proteins that act as catalysts in living organisms. They regulate the rate at which chemical reactions proceed. The enzymes are not themselves altered in the process. The enzymes I was studying are proteins that take apart complex carbohydrates into simple sugars for recycling in the lysosome. My lab was interested in investigating how these enzymes were delivered to the lysosome. We knew that the enzymes were made in the rough ER and that they were processed in the Golgi complex. But we didn't know how they got shipped from the Golgi to the lysosome. A few years earlier, Barbara Pierce, a British biologist, had described cell transport vesicles called coated vesicles, and I wondered if these vesicles might be involved in transporting lysosomal enzymes to the lysosome. My first graduate student, Corinne, and I set out to look for the presence of lysosomal enzymes inside these vesicles. Here's my lab group in the early 80s. Corinne is on the left. Corinne followed the published method for purifying coated vesicles, and she was able to show the presence of lysosomal enzymes inside her purified coated vesicles. This was around 1983, and we had just begun to use a technique called negative staining to look at the coated vesicles under the electron microscope. Before we learned negative staining, we had to purify and collect the vesicles, treat them with a fixative and a stain, embed them in plastic, and cut razor-thin slices with a microtome before we were able to view them under the electron microscope. This procedure was very slow and laborious. Remember, I used it to show you what a sample of my blood looks like. The negative stain procedure is a far easier method than the one we were using. You put a purified sample on a carbon-coated grid and follow with a heavy metal stain. We were able to get very nice images of the coated vesicles. Here's a picture of some negatively stained coated vesicles. Notice the soccer ball-like appearance. I remember one day, while viewing some of Corinne's micrographs, we noticed an unusual particle that was the same size as the smaller coated vesicles. This particle, circled here in red, however, was somewhat different in its morphology. Morphology is just a fancy way of referring to the size, shape, and structure of something. Notice it wasn't round, it was ovoid, and it didn't have the soccer ball-like surface that coated vesicles had. I remember asking Corinne about the particle. However, they were unrelated to her project, so she did not attempt to follow up on these unusual structures, and the new observation went uninvestigated. You know, this could have been the end of the story, and my career may have gone differently, had it not been for a new addition to my lab who moved our research in an entirely new direction. I'm Lenny Rome, the Vault Guy. I hope you join me on my next video so I can tell you more about finding vaults and learning about this incredible particle.